Biochemistry Module Lecture 6, DNA and Biotechnology. We'll start this lecture off by talking about DNA structure. DNA is comprised of nucleotides. These are comprised of nucleosides with 1 to 3 phosphate groups added. The bases that you'll notice are deoxyribose in DNA and ribose in RNA. These are how these two compounds get their name: deoxyribonucleic acid and simply ribonucleic acid. There are 5 bases that you have to talk about here: adenine, cytosine, guanine, thymine, and uracil. In RNA, you're going to notice that thymine is replaced by uracil, so that's how you keep the two molecules separate. Below is a structural categorization of purines and pyrimidines. In the next slide, we'll go into ways to remember these. The established structure of DNA was discovered by Watson and Crick several years ago, which is why we use the Watson and Crick model when we model DNA. It simply states that the backbone is always read 5' to 3'. Strands run next to each other in anti-parallel fashion. What that means is that if one side is running from 5 to 3, the other side has to be running in the opposite direction, like so, when they pair up. That's what it means to be anti-parallel. Purines pair with pyrimidines, and RNA, as I said earlier, has U instead of T, uracil replacing thymine. Chargaff's rule simply states that purines and pyrimidines are equal in number in a DNA molecule, and this is because of the base pairing. Things that can denature DNA are the following: heat, alkaline pHs or basic pHs, chemicals, formaldehyde and urea being the most common. And obviously, if you remove these denature conditions, you can probably reanneal the DNA if you try hard enough. That's a lot to take in, but how can we remember what bases go where and what they're comprised of? This will definitely be tested on the MCAT, so there's a lot of ways you can remember it, but the best way I've come up with is to use pure as gold and cut the pie. So I'll go ahead and list those out. There's pure as gold and cut the pie. So what do these mean? Well, purines are adenine and guanine, pure as gold, and CUT are listed as, obviously, pyrimidines. Cut the pie. And for both of these cases, well, I see 3 words. And this is to show that the GC has 3 bonds. So when you pair these two together, you're going to see that AT, or uracil in the case of DNA, and GC bonds are going to pair together. So for AT, you've got 2 bonds going across, whereas for GC, you have 3. So GIC, 3 words. So the way I like to remember these is simply pure as gold, cut the pie. I see 3 words. So again, purines, pure as gold, pure, AG, cut the pie, CUT, pie, pyrimidines, and GIC words, G and C have 3 bonds. That's the easiest way to remember this from what I found, and these will definitely be topics you'll see on test day. Here's a diagram listing out the structure of what DNA looks like via the Watson and Crick models. I know that in a lot of your genetics classes you talked about major and minor groups, but those sort of topics won't come up on test day. As you can see, the strands are running anti-parallel to each other, simply stating one side goes 5' to 3 and the other is mirrored 3' to 5. That's it for DNA structure. Let's go ahead and look at how they're organized in eukaryotic chromosomes. The human chromosome is listed to have 46 chromosomes, 23 from each parent. DNA is also wound around histone proteins to form the nucleosome, and this is organized into chromatin. This is DNA and its associated histones, and there's 2 types: heterochromatin, which is dense and transcriptionally silent, and euchromatin, which is less dense and transcriptionally active. If it's dense, that means that it's wound in tightly, meaning that mechanisms for transcription cannot get in, whereas euchromatin is less wound up, meaning that these sort of transcriptional mechanisms can get in. The way I like to remember this is that euchromatin is useful. There are some key components to chromosomes that we have to talk about, and there's two in particular that the MCAT likes to test. There are telomeres, which are the ends of the chromosomes. Since they're the ends, they have high G and C content to prevent unraveling. If you remember, G and C bonds, base bonds, have 3 bonds in between them. So they are more stable than the AT bonds. Because of this, if you look at gene sequences, you can often tell where telomeres are, and centromeres as a matter of fact, based on GC content. They slightly shorten during replication, and this can be slightly reversed by the telomerase enzyme, but every replication you have some depreciation here. There's a lot of theories that say this is where aging comes from, because we have a fixed amount of replications within our sequence. Centromeres are the middle, and this is what holds sister chromatids together until they separate during anaphase and mitosis. Like telomeres, they have high GC content. If we were to draw out what a chromosome looks like, so if I were to have a chromosome like so, the centromere would be here, and the telomeres are going to be the ends, so these structures right here. Like I said earlier, these telomeres will shorten, and the centromeres are what hold the 2 sister chromatids together. During mitosis, this is where the spindles will attach to pull these apart. When it comes to DNA replication, there are a couple proteins that we have to talk about. The replication complex is noted as a mechanism that involves helicase, polymerase, topoisomerase, and some ligase as well. Helicase unwinds DNA, which yields a replication fork. For prokaryotes, this is a circular chromosome with one origin, whereas eukaryotes have a linear chromosome with multiple. As you might guess, it's possible for replication forks to go in 2 separate directions. So if I were to have a DNA like so, if a replication fork were to occur, you would find that it could potentially travel in both directions. There are single-stranded DNA binding proteins, and this is simply to keep the DNA from reannealing while this complex goes through. Topoisomerase 2 relieves the torsional strain from unwinding the DNA. This is done via supercoiling. If you've ever twisted up a rope and tried to twist it further as you pulled along it, you would know why this sort of torsional relief is necessary. DNA polymerases will synthesize new DNA strands along the single-stranded DNA. These polymerases will read 3 to 5, however, DNA is always synthesized 5 to 3, and the same will go for RNA as well during transcription. The leading strand has 1 primer and is continuous, whereas the lagging strand has multiple primers and is discontinuous since it's reading in the opposite direction. These fragments are called Okazaki fragments. The primers noted are always going to be RNA, so make sure you're aware of that, and these primers are later removed and replaced with DNA. DNA ligase is what fuses the DNA strands together after the replication is complete. If you remember from lecture 2, you'll remember that ligase enzymes will join 2 large molecules together. So here's one at work. Here's a diagram listing what this sort of complex is going to look like. If you see something that's discontinuous, you can obviously note that that is going to be the lagging strand, whereas the leading strand is going to have a continuous setup. DNA repair occurs in several different mechanisms, and we'll go into those in this little portion. Before we talk about ways that DNA replays itself, we need to talk about what constitutes a mutagenic gene. There are 2 genes in particular that can cause errors in replication. Oncogenes are mutations in proto-oncogenes, and they promote cell cycling and could possibly lead to cancer if uncontrolled. Cancer is noted as the unchecked cell proliferation that can spread throughout, and this can happen in any cellular environment. If a cell loses its ability to regulate its replication, it's going to create cancerous effects. Tumor suppressor genes they code for proteins that reduce cell cycling or promote DNA repair. Mutations here can also lead to cancer. Obviously, if our suppressor gene has an error, it's not going to be able to suppress the formation of tumors, leading to cancerous states as well. There are 3 repair mechanisms used in the cell, and the first being mismatch repair. This replaces errors after scanning during the replication process, and this usually does a pretty good job. However, if replication completes and there are still errors, there are 2 other types of repair mechanisms that the cell will employ. Nucleotide excision repair fixes helical deformation lesions of DNA. This utilizes a cut and patch process via an excision endonuclease. The most classic example of this is a thymine dimer. A thymine dimer forms when you have two thymines next to each other that bond together. So if I were to have something like this and have two strands, a thymine dimer, especially in the presence of UV radiation, is going to bind to itself instead of where it needs to. What's simply going to happen is an endonuclease is going to come in, cut this out, and replace it with the proper TT bases. Base excision repair fixes non-deforming lesions of the DNA helix. This removes the erroneous base, leaving an apyrantic or apyrimidinic site, AP site, with AP endonuclease then removing the entire damaged sequence. The AP endonuclease then fills the AP site with the correct base. So if there is some sort of error, you'll often see these come in and just cut it out and replace it, similar to a nucleotide excision repair. However, nucleotide excision repair often uses the structural deformities to locate these errors, whereas a base excision repair does not need such a thing. You'll notice that with a lot of genetic engineering techniques, AP endonucleases and these types of repair mechanisms in the cell are used to incorporate new DNA into the sequence of the host. Speaking of these biotechnologies, we'll go into some recombinant DNA and biotechnology in the next portion of this lecture. We'll start first with some basics. Recombinant DNA is basically any DNA that is composed of nucleotides from different sources, meaning that there was some sort of foreign DNA introduced into a host. DNA sequencing is what is used to determine these base orders of DNA, and this is done by things such as the Edman degradation, which we talked about in a previous lecture. DNA libraries are noted as stores of known DNA sequences, and these can be trillions upon trillions of bases long. Genomic libraries contain both coding and non-coding fragments of DNA, so this is what you would consider to be just the entirety of these genomes, whereas cDNA contains only exons, or express DNA fragments. So, long story short, genomic libraries are the entire library, whereas the cDNA libraries are simply the things that we want. Hybridizations are noted as the joining of complementary base pair sequences. The cell naturally does this, but when it comes to genetic engineering, we can use these intracellular mechanisms to incorporate other DNA sequences within. You'll often see that viruses, such as retroviruses, will often use similar mechanisms to incorporate themselves. Polymerase chain reactions, or PCR for short, is the automated process of creating multiple copies of a DNA sequence. So what happens is if you have one sequence, it's copied over and over again. And this is done exponentially. This can go indefinitely, and this is a good way to replicate multiple copies of a sequence. DNA can also be separated by gel electrophoresis, which is another thing that we talked about in Lecture 3 when we talked about protein analysis and isolation. Southern blotting is used to detect the presence and quantity of various DNA strands in a sample. This is done by probing DNA samples with single-stranded DNA to look for a specific sequence. Obviously, if a sequence is complementary to the probe, it's going to bind to it, therefore locating the sequences that we want. Cloning is noted as the introduction of a DNA fragment into a vector plasmid into a specific region via a restriction enzyme. Restriction enzymes being things that incorporate DNA within itself. The vector is then introduced into a bacteria, which then expresses the desired trait. This is oftentimes how we augment certain cells to exhibit traits that are not natural to it. Below is a diagram of how this process is done using a plasmid vector in bacterium. There are ways to genetically modify organisms, and these are sort of a novel treatment that is sort of creeping its way into medicine. Gene therapy is a method of curing these genetic deficiencies or disorders by introducing foreign functional genes using viruses. These viral vectors insert the functional gene into the host genome to alter the pre-existing genome. Therefore, if the host has a faulty trait or gene, these viral vectors will introduce the proper gene and splice itself in, therefore giving the host a functional gene that allows for proper function. Transgenic mice are created by using these types of mechanisms. They're created by introducing a gene of interest into a germline of embryonic stem cells, thus forming chimeras, which are organisms that contain cells from different lineages. They can be selectively mated to derive a desired transgene, and knockout mice are noted as having a gene of interest deleted. using these sorts of modifications, we can specifically test what these genes do and how their expression is going to affect the overall organism. Gene therapy is currently a treatment option that people are looking at for things such as sickle cell anemia and things like HIV or other viral-based illnesses. This is because those illnesses are directly derived from genetic expression. Therefore, changing the genome can obviously potentially treat these illnesses. This does raise a lot of substantial ethical concerns, though. When it comes to genetic engineering, it is an immensely controversial technology. There's a lot of questions that it brings up, and the MCAT will often have passages in your CAR section that are going to try to address these things. For starters, should we even be doing these sorts of augmentations? And if we do, who has the authority to allow this? I mean, we're talking about the human genome. Who has the authority to say yes or no regarding the alterations of our genome as a species? Also, when is it acceptable to do this? Where do you draw the line when it comes to, do I want to adjust genes or not? Do we use it simply for life-threatening genes or life-threatening illnesses? Or do we allow parents to genetically augment their children so that they exhibit the traits that they want? Do we understand the consequences fully, as a matter of fact? Science is kind of known for being a little bit short-term with its threat and risk analysis. Altering the genome now could have positive benefits in the current moment or for a couple generations, but how is this going to affect us further on? Do we even have the tools to understand this? These are questions that the MCAT will often ask you, and these are questions you should probably be asking yourself too when it comes to these sorts of technologies. We'll finish up our discussion of DNA and biotechnology there. In the next lecture, we're going to go into the second genetic code, and that being RNA. We'll see you then.